Thanks, Tony, for the introduction, and I'd like to thank the Alison and the organising committee for giving me the opportunity to present some of our work. I've just some disclosures of some companies that we have had some research funding from, but I'm not presenting any of that work today. I'm really pleased to be able to give this talk because I really would like to thank the IBTS, who have facilitated all of my research for 27 years. And it's not even now and again, it has been a constant thing. We have been getting Buffy coat packs from the IBTS. As you see, we work with cells that are very rare, so we need lots of blood in order to get enough cells to use. We used to, even at the beginning, I used to cycle over to Mespil Road to use the irradiator to irradiate our cells, and I'll show you why we did that in a minute. So all of the work started when I was a postdoc in the lab of Professor Cliona O'Farrelly, and we were interested in the liver being a tolerogenic organ, one that any antigens that first are encountered in the liver, they induce immune tolerance rather than an actual immune response. So we were very interested in this, and we were taking bits of donor liver before transplantation, so it was healthy liver, to be able to look for the presence of lymphocytes. And it was originally thought around that time that there are only lymphocytes in the liver if it's a diseased liver, if it's got an infection. But we found that there are as many lymphocytes in the liver as there are in the blood. So we've been isolating these and trying to grow them up and phenotypically characterizing them. And what we found was that there's very unusual lymphocytes in the liver. A lot of them had characteristics of natural killer cells, that really set us off. That was the original finding of lymphocytes, which we then looked in lots of other organs and in blood. And that's what I've been working on pretty much since. And there's several different types, but I'm only going to talk about one of them today. And it was this by my first PhD student who found natural killer T cells in the liver. So that's really the subject of my talk. But before, I'd like to just mention that at that time, it was generally known that the immune system is involved in rejecting tumors. But there was really no proof. There was absolutely no proof. And I think this is one of the first papers. So it's only, this was just looking at mice that would get that would spontaneously develop cancer. And if you knock out genes that are encoding lymphocytes, for example, RAG2, interferon gamma receptor, STAT1, and this is a double knockout, you increase the chance of getting cancer, suggesting that lymphocytes are involved in the immune response to cancer. And since then, there have been loads of studies. So here are all the different knockout mice that have been made in various models of cancer. And you can see any of these cells led to if you knock out any of these molecules, which lead to the knockout of cells in many cases, you increase the risk of developing cancer. And all these are all cytotoxic cells or molecules that are involved in cytotoxicity. So very, very strong evidence that cytotoxic lymphocytes are essential for eliminating tumors in mice. And in humans as well, the presence of tumor-infiltrating lymphocytes is a good sign. It's associated with a favorable prognosis. And certainly we've been able to expand tumor antigen-specific cells. So it's now well understood that lymphocytes, in particular NK cells and cytotoxic T cells, they can kill tumor cells in vitro by making physical contact, releasing toxic chemicals that will kill the cell, and these would include perforin and granzyme, as well as the ability to ligate death receptors, leading to apoptosis of the tumor cell. And this finding has led 20 years later to the actual immunotherapies that involve the use of lymphocytes. The checkpoint inhibitor drugs that can remove inhibitory signals from lymphocytes, and also the cellular therapies, which we're particularly interested. You can take lymphocytes that are engineered to express chimeric antigen receptors to enable them to target them to the tumor, or we can engineer them with T-cell receptors that are specific for tumor antigens, or you can go in an unbiased way and just take tumor-infiltrating lymphocytes, grow them up in vitro, and transfuse them back to the patient. And CAR T-cell therapy actually is becoming a reality here in Dublin. So we know that cytotoxic T cells and natural killer cells are both able to kill tumor cells and both mediate tumor rejection, both by cytotoxic means as well as by releasing interferon gamma and tumor necrosis factor and other cytokines that will activate other cells of the immune system. But what I'd like to do is talk about innate T cells, and this is another type of cell that seems to be very involved in tumors. Innate T cells are T cells that recognize non-peptide antigens, and they are not restricted by MHC molecules. So they have non-MHC antigen presenting molecules presenting non-peptide antigens, and the cells that recognize them very often have semi-invariant T cell receptors that don't undergo the same diversity generation as normal T cells. And the cells that we've been looking at are these. There's gamma delta T cells, and we've been looking at the three different subsets in humans. There's mucosa-associated invariant T cells, and there's natural killer T cells. And it's only the natural killer T cells I'm going to show you today. This just shows a typical flow cytometry dot plot of looking at T cells, shown in here, and the CD3 positive cells, and natural killer T cells expressing this invariant V alpha 24J alpha 18 T cell receptor. They account for less than 1% in just about all humans, but they are of great interest because that is quite high for a single antigen specificity of a T cell. They express this invariant T cell receptor as well as natural killer cell stimulatory and inhibitory receptors. So they have properties of both NK cells and T cells, and hence their name. And this is two papers that came out in 1997, and this was before natural killer T cells had been studied in humans. But it was found that the equivalent cell in mice, which is called the V alpha 14 NKT cell, could be activated by this molecule. And this is a molecule, alpha galactosyl ceramide, which was used as a screen in a model of murine cancer. And it was found to reject tumors. If you just inject that into the mice, it could prevent as well as reverse tumor development. And these were two papers by the same group back to back that showed this. So this led us to want to look at these in humans. And just to revise, this is the NKT cell. It's called the invariant NKT cell because it's got this semi-invariant T cell receptor. It's an invariant alpha chain, but there's a bit of variability in the beta chain. And it recognizes CD1D presenting glycolipid antigens, and this is the first lipid antigen that was discovered. And CD1D is very, very similar to an MHC class 1 molecule. Both consist of an alpha chain with 3 extracellular domains, shown here in red, and they're both covalent, non-covalently associated with the beta-2 microglobulin. And the only real difference is the antigen binding groove on an MHC class 1 molecule binds peptides, whereas on the CD1D molecule, it has a very large hydrophobic groove, enabling the burying of these acyl chain hydrocarbon chains deep into the molecule. And the galactose, the glyco part of the molecule, sticks out for interaction with the T cell receptor. So several studies in mice have shown that NKT cells are essential for mediating tumor rejection. And this seems to be not just that they can be very early producers of interferon gamma and thereby activating NK cells and cytotoxic T cells. And we know that knockout mice lacking NKT cells don't have this property. They are not able to reject the tumors. And the protection against the tumors can be transferred by the adoptive transfer of invariant NKT cells from one mouse to another, showing that these do indeed have a very important role, at least in mice. And the way they seem to work is they are directly cytotoxic, but they also release cytokines which will result in the activation of other cells, typically natural killer cells and cytotoxic T cells, which infiltrate the tumor and lead to tumor cell death. And so we decided we wanted to look at these cells in humans. And there are much less of these cells in humans, but we did a lot of studies looking at using flow cytometry, looking at the frequency and the function of invariant natural killer T cells in various cancers. And this just shows, several different cancers: colorectal, liver, esophageal, lung cancer, chronic lymphocytic leukemia. And in all cases, the numbers of lymphocytes in the blood are decreased quite significantly. And they're in low numbers anyway. There's about, as I mentioned, less than 1% in human blood in healthy donors, and that's decreased in cancers. Whether that is because they go to the tumor, we don't know, but we don't think so. This just shows actually the numbers of cells in liver or in adipose tissue or in bronchoalveolar fluid. So there seems to be a deficiency of NKT cells in all tumors. And this has led several groups to carry out clinical trials targeting NKT cells. First of all, the first ones were injecting alpha-gal-CIR or alpha-galactosalceramide into the patients. That was quite safe and well tolerated, but it didn't lead to any elimination of the tumor. So another strategy was to isolate the NKT cells, grow them up in vitro using the alpha-gal-CIR antigen and IL-2, and then adoptively transfer them back to the patient. And there's been many clinical trials using this method. And the third method was to isolate peripheral blood mononuclear cells from the patient, isolate the monocytes and mature them into dendritic cells in vitro, load them with alpha-gal-CIR, and then adoptively transfer them back to the patient. And there's even been several clinical trials using a combination of NKT cells and alpha-gal-CIR activated dendritic cells. So how did this all work? Well, nothing has been licensed yet. However, the promise is still there, and there's these many studies have really generated the proof of principle for harnessing NKT cells to prevent cancer in humans. So although nothing's been licensed, the cancer trials are still going, and I would be very confident that they'll be licensed for some tumors in the next few years. But why, so far, have they not been successful? And that was really what we set out to do. First of all, we wanted to isolate them and look at them. But innate T cells and NKT cells account for less than 1% of cells in the blood. So we need large volumes of cells. Let's just say this is the one NKT cell, and we want to grow that out. We need a large volume of blood, which we would get from the IBTS, and we would grow it in the presence of antigen and irradiated feeder cells. These are cells that would give non-specific set of goodies to help the cells grow. And you can expand them very dramatically. This again, we got these irradiated feeder cells from the Irish Blood Transfusion Centre. And another way of doing it, which we found was even easier, we could just isolate that one NKT cell and then grow it with a mitogen such as phytohemagglutinin. And we could get, using these methods, we could get tens of millions of NKT cells fairly pure. To just show you a flow cytometry dot plot. So this process would take about a month to grow. And the cells actually are surprisingly stable. They would last for a couple of months on a 96-well plate. We'd just feed them once a week. So we had the cells to work with. And in order to work with them, we would co-culture them with cells expressing CD1D and add the glycolipid. We have various transfectant cells expressing CD1D. We've also got, we can co-culture them with tumor cell lines or primary tumor cells, or dendritic cells or B cells, because we were really interested in the basic function. And what I'd like to really tell you is what we've done, co-culturing expanded NKT cells with B cells, just to see that that's really the focus I'm going to look at. And when they were doing that, we see what are the activities. Cytotoxicity, cytokine secretion, differentiation, antigen presentation, and antibody production were all looked at. So I'd like to just show you some of the results, and this is a result from a PhD student, Vinnie O'Reilly, and he was able to show that when you stimulate NKT cells with alpha-gal SEER, the antigen I mentioned, and then you gate on, there's three populations. There's the CD4s, the CD8s, and the double negatives. And consistently, the CD4s are much better at producing the Th2 cytokine in IL-4, whereas the CD8s are much better at producing the Th1 cytokine interferon gamma. So the CD8s would be expected to be the better anti-tumor effectors. And at this time, there was some evidence that CD4 NKT cells might actually promote tumor growth rather than inhibiting it. And Vinnie also looked at the cytotoxic activity of CD8s, CD4s, and double negatives and found, again, the CD8s were much more cytotoxic. Now, a very interesting about NKT cells is normally T cells would be a Th1 cell or a Th2 cell or a Th17 cell, and you don't get mixed responses. But with NKT cells, as I showed you in that flow plot, you can have a mixed Th1, Th2 response. But we're seeing that the Th1 response seems to be more concentrated in the CD8s, and the Th2 response is more CD4s. However, when you look at the frequencies of these cells in human blood, we see the CD8s are the most popular, but when we expand them, the CD4s grow much better. And this is the method that's been used for just about all the clinical trials. So it suggests that the wrong subsets of iNKT cells were being used to treat cancers, and that might be a reason for the lack of efficacy. Mostly 20 years ago now, this paper came out in Nature, and it was a new glycolipid. At that stage, this was the only glycolipid known to activate NKT cells until this paper came out, and it was found, the glycolipid is called OCH, and it's just got a few modifications, but it induced a Th2 response. And this is very exciting, because if we can make it be a Th1 cell or a Th2 cell, then we can tweak the activities of the cells in order to do what we want them to do. And around that time, another paper came out with this alpha-C glycoside, and it only induced the production of Th1 cytokines like interferon gamma. And in mice, activation of this cell with this glycolipid, which actually only differs by one atom, this carbon replaces an oxygen, that had a 10,000 times greater anti-metastatic effect in mice than the original alpha-gal SEER. The only problem is it did not activate human NKT cells. So that was the first thing I wrote to the guy who produced that, and he gave me the lipid, and it didn't work at all in human cells. So there are a lot of big differences. But so what we did instead, we got a collaboration with this guy, Changming Zhu, in UCD. He's a synthetic chemist, and he was interested in making variants of alpha-gal SEER. And what he did was he made a thioglycoside. He just replaced this oxygen with a sulfur to make a thioglycoside analog of alpha-gal SEER. And we tested this. This was the work of Andy Hogan, who was another PhD student, and he compared alpha-gal SEER and the thioglycoside analog, and he found that both of them stimulated cytotoxicity, killing of tumor cells, and both of them stimulated interferon gamma production by NKT cells. Not really any different from alpha-gal SEER. It had a very, very similar effect. So we hadn't really come up with anything better, except maybe a slightly more stable compound in human systems. But Changming went on to make more variants of this, and he came up with many variants that we tested. But this is the one we are particularly interested in. It's got 2 galactose residues, and then it's got the long chain acyl chains. And this work was done by Ashanti Mello, who tested loads and loads of glycolipids to see how were they capable of activating NK T cells. And this one was one of exciting because it induced CD107A, which is a marker of cytotoxicity. It induced interferon gamma, but it didn't induce IL-4 production. So this was a biased response towards a Th1 response, which is what we want in order to cure a cancer. Interestingly, it only activated the CD8 NKT cells. So it's activating the cells that we want to activate, and we don't want to activate CD4 T cells CD4 NKT cells, because we know that they are capable of, in some cases, prolonging or promoting tumor growth. So we're not really there yet, because the activities are still not as strong as alpha-Gal-Cr, but they are the right types of activities. So Changming is still making more lipids for us to test. I'd like to now just go to a bit of work for the rest of this talk, is to just look at interactions of NKT cells with B cells. And this was something we were particularly interested in. We looked at interactions with dendritic cells and with other cells of the immune system. But I'm just going to show you the work with NKT cells. And this work was done by Grace Zheng, another PhD student. And Grace took B cells, co-cultured them with NKT cells, added various glycolipids, and then found that the B cells matured in response to this into plasma cells that were capable of releasing antibodies and producing cytokines. And this is really like what a follicular helper T cell would do in the lymph node, showing that NKT cells have properties of follicular helper T cells. They're able to give help to B cells to produce antibodies. And Grace found that she could get considerable amounts of IgM, IgA, and IgG when B cells were co-cultured with NKT cells. And this did not occur if they were co-cultured with any other cells that we used. I think we mainly used total PBMCs. And Grace also noticed that the NKT cells induce phenotypic changes in B cells, specifically the upregulation of CD40, CD83, and CD86. And these are molecules found on antigen-presenting cells. And we know that in some cases, B cells can present antigen to T cells. So Grace looked to see could they present tetanus toxoid and hepatitis B surface antigen and various superantigens to T cells. And she did this by using an assay where she'd label the conventional T cells. These were autologous T cells, only possible because we can take T cells and NKT cells from the same donor if you can get large volumes of blood, as we were able to get from the IBTS. And these were labeled with cell trace violet. It's a label that once the T cell divides, it will divide the amount of dye into 2 daughter cells, and as the dye dilutes, you get different peaks on the flow cytometer. So here we have T cells alone. They haven't divided, so all of them have a high concentration of cell trace violet. If you give them a mitogen like PHA, they will give, so each peak is a cell division. When we gave them B cells, we could see, again, cell division, suggesting that the B cells are inducing proliferation of the T cells. However, when we added iNKT cells, we were very surprised that we blocked that proliferation. So it looks like iNKT cells are actually inhibiting antigen presentation by B cells. I should note that when I say iNKT cells, this was purified CD4 positive iNKT cells. This didn't work for the CD8 NKT cells. And then Grace also noticed that when she co-cultured B cells with CD4 positive iNKT cells, it induced a phenotype of CD5 and CD1D, which is a phenotype that had been just around that time a phenotype that characterizes B-reg cells, or regulatory B cells. Regulatory B cells are a bit like regulatory T cells in that they release IL-10. Some groups call them B10 cells. And we found that, yes, these B cells did produce IL-10, and they could produce IL-10 when they're co-cultured with CD4 NK T cells in the presence or in the absence of alpha-gal CIR. So it looks like these didn't need to be activated in order to produce B cell, to induce the differentiation of B cells into B-reg cells. And the last question Grace asked was, can NK T cells kill B cells? So she took NK T cells and autologous B cells, co-cultured them, and measured the expression of CD107A, the degranulation marker, and found indeed they do kill B cells. Although they don't kill B cells to the same degree as they kill tumor cells, or as this graph shows, dendritic cells, they're quite weak at killing them. But this was enough to make us suggest, do they kill BCLL cells? And this was work done by another PhD student that was co-supervised by myself and Tony McGilligan. And Yasmin set out to find out, can NK T cells kill autologous chronic lymphocyte, CLL cells? And she looked at a few things, and one of the first things she found that they didn't kill very well. And one reason might be, well, CLL cells, or B cells in general, from patients, had lower levels of CD1D on their cell surface. And this is showing a lower percentage of B cells express CD1D, and of the ones that did, the intensity of expression was very low. And this was both in the CLL cells expressing CD5, as well as the healthy B cells that are negative for CD5. So CD1D is downregulated on B cells in CLL patients, suggesting that if we could upregulate CD1D, we might be able to induce them to be killed by NK T cells. And around that time, we found this paper that said CD1D is upregulated on B cells by retinoic acid. So we got some retinoic acid to see if that could be done with CLL cells, and indeed it could. We were able to induce, at very low concentrations of retinoic acid, we were able to induce CD1D to quite high levels on B cells from both patients and from healthy donors. And we then co-cultured them with, or I should say, Yasmin co-cultured these cells with NK T cells, expanded from blood packs, and found that they killed, but only the CD8s were capable of killing when they were added to B cells. Sorry, I should say, when we added all-trans retinoic acid, or its analog AM580. So we found we could, retinoic acid could prime B cells for killing by invariant NK T cells. And we also tried other agents that might upregulate CD1D, and this was a collaboration with Steve Gray, in which we both co-supervised this PhD student, Irish Dockery. And we used, or she used, epigenetic modifiers to try and induce CD1D. And sure enough, we were able to induce CD1D on cells that appeared to be, this is A549 lung cancer cell line. We were able to induce CD1D even on cells that seemed to be absent for CD1D. And this made these cells susceptible to killing by NK T cells. You can see here, for increasing concentrations of two different glycolipids known to activate, increasing concentrations of the DAC, which is the epigenetic modifying agent, and in the presence of glycolipids, you could see a dose-dependent activation of cytotoxicity by these cells, suggesting that we are finding another way to make cells visible to NK T cells in order to kill them. And finally, I'd just like to say we're taking this work a bit further, and this is the work of Hayley Foy-Stones, who's here today and had a poster last night. And she's looking at NK T cells and other innate T cells post-allogeneic stem cell transplantation and post-CAR T-cell therapies at many times. And the hope is that we will find something interesting about NKT cells. And there is a little bit of evidence in the literature that NKT cell reconstitution in patients following allogeneic stem cell therapy leads to better outcomes. So this work is just starting out. So to summarize, we found that NKT cells kill CD1d positive tumor cells and promote tumor cell killing by other cells of the immune system. We found that CD8 NKT cells are by far the most potent anti-tumor cells, whereas the CD4s may promote tumor growth. And the anti-tumor activities of CD8 positive NKT cells can be enhanced by stimulating them with various glycolipid analogues. And we also found that the activities can also be enhanced by inducing CD1d expression using retinoic acid or epigenetic modifying reagents. And Grace's work was how NKT cells are capable of controlling B-cell responses. They promote antigen secretion, they inhibit antibody secretion, they inhibit antigen presentation to conventional T cells, they induce the differentiation into B-reg cells, and they also kill B cells. So we've got lots and lots of functions of NKT cells, which we hope we can selectively activate in order to treat cancer. And I should mention that these cells are also being used to try and treat autoimmune disease and infectious disease because of their ability to do so many different things. So I believe that there's a future in NKT cells to be targeted for cancer immunotherapy. And the main reasons are they can be easily expanded, as I mentioned, in vitro. We can even do this now with quite small amounts of blood, for example, from patients. And there are multiple effector functions, most specifically the rapid tumor killing and the rapid cytokine secretion. But also, these can be tweaked to do what we want. And for example, we can choose the type, the class of NKT cell. We can choose the type of antigen-presenting cell. We can choose various different glycolipids to activate the NKT cell. And we can actually also, we've done quite a lot of work looking at the ability to bias a response of NKT cells via cytokines. And finally, we can also activate NKT cells or maybe even co-stimulate them through other receptors, the receptors typically found on natural killer cells, such as NKG2D in particular. And NKT cells also, their main role appears to be to control other cells of the immune system at a very early stage of an immune response. And this just shows the many physical interactions of NKT cells. They interact with B cells, macrophages, neutrophils, myeloid-derived suppressor cells, dendritic cells. And as well, they can activate many other cells just by the release of cytokines. NKT cells have the promise of being used as allogeneic therapies because since they're not MHC restricted, they're unlikely to cause graft versus host disease. So this means we don't need expensive personalized treatments. And finally, they can be engineered as CAR T cells. And there have been some studies already that have generated CAR T cells. So in this way, we're able to introduce ways to target them to the tumors while retaining their anti-tumor properties. And I should mention, I've only really talked today about NKT cells. There are many other innate T cells, the gamma delta T cells and the mate cells, that are also being tested in clinical trials for cancer. So there's a great future, I think, in innate T cells in the cancer field in the future. And finally, I'd just like to acknowledge the people that have done all this work, the mainly PhD students. We've got several collaborators. And of course, I'd like to mention the Irish Blood Transfusion Centre, which has made all of this work possible. And every green cell that I showed you in all my slides, they all came from blood packs from the IBTS. And thank you very much for listening.
Thank you, Derek, for that great talk. Actually, I think we were well stimulated by that. This is open for questions. Professor Madriel. Have you got a, yeah, up at the front too.
Anyway, I can speak loud. Fascinating talk. Congratulations, Derek. I was really impressed by you. It's a long time effort work. We met a long time ago in Stanford, and you nearly come to where I was at with the Stanford with Peter Parham. One of the most fascinating points that you highlight is the interaction between the CD4 and CD8 different actions, one promoting the tumor and the other one sort of and eliminating it. The question is how the CD4 actually promote the actual tumor, because that might be something in vivo that might be quite critical. Yep. Is that mediated because the CD8 in some way, the CD4 eliminate the actions of the CD4 or other activated T cells that might be able to eliminate the tumor? So what is the mechanism by which the CD4 prevents the NK of the
We really don't know the mechanism, and it's hard to look. In mice, things are different because mice have CD4 and double negative NKT cells. They don't have CD8s at all. So we're looking at something very different from what we've seen in mice. And we don't know how, I don't even know why NKT cells have CD4 and CD8 since they're not MHC restricted cells anyway. But we notice that they do. We know that CD4 NKT cells are very often found in tumors that do badly. So we need the influx, the tumor infiltration of CD8 NKT cells seems to be the most important finding. So I cannot answer your question, and it's the burning question for decades that we've been wanting to know. What is CD4 and CD8 doing on these cells? All we know now is they're markers for cells with slightly different but overlapping activities.
But you haven't tested if the actual presence of the CD4 prevents the activity of the CD8 or the activation of the CD8?
No, we haven't done that.
I think that would be...
What we have done most of our work with mixed populations of NKT cells. When we did sort them, we did try to see which are the best killers and all, but we didn't really see any specific killing.
Did you look for MIC-A or MIC-B expression on those?
We didn't look, no, but we do know that they have the receptor for MIC-A and MIC-B. NKG2D is found on pretty much all NKT cells.
Thank you very much.
Thank you.
Hi, thank you very much for the lovely talk. Just two stupid questions. What's about the microenvironment, the liver microenvironment? Because you were mentioning that, if I correctly understood, they're mostly found in the liver. Could be anything there that, kind of Kupfer cells, something that might co-stimulate, the microenvironment of the liver might be something there. And then the other little question is about, if you could comment about autoimmunity. You mentioned that they might have a role in autoimmunity.
Yes, the first question, I fully agree that when you take probably any cell out of its environment, you change the properties. So, and we do know that Kupfer cells are able to activate T cells in the liver. They are actually very, very good tolerogenic antigen-presenting cells, as well as many other antigen-presenting cells in the liver. So you're absolutely right that we might be seeing something different in vitro compared with in vivo. And your other question was about autoimmunity, something we haven't looked at, but there's been many studies that have found that NKT cells can prevent the development of experimental autoimmune encephalomyelitis, the murine model of multiple sclerosis. NKT cells play a role in type 1 diabetes, in mice again, and in rheumatoid arthritis. There are many roles. What we think really, NKT cells are involved in just about all immune responses at a very early stage, and at that stage when they can polarize an immune response towards one direction or another, like a Th1 versus a Th2.
Probably a difficult question to finish up with there, Derek. I mean, the IBTS, as you said, has been instrumental in driving this research with the for isolation, the NKT cells. What further interactions with the IBTS could you envisage, sort of to support your research or to support collaboration between your research group and the IBTS?
Well, the wonderful thing is that we have this new collaboration, and that is with many people in this room looking at allogeneic stem cell therapy. So I'm delighted to be able to interact with the IBTS really much more directly rather than just obtaining buffy coat packs. So I'm hoping to do lots of stuff in the future.
And in 2 years' time, we'll hear all about it. Yeah. Thank you very much, Derek.
Thank you.